Here we go with part 3. So this is another part on multiple choice questions. So paper 1 questions on membrane structure. So let's just keep going. So what part of the plasma membrane is fluid, allowing the movement of proteins in accordance with the fluid mosaic model? So what part of this membrane is fluid? So we know the part that is fluid is these phospholipids. The phospholipid tails are very bendy, and they basically make the membrane quite weak and quite fluid. Why is it not A or D or C? Because these are proteins. So C is a protein. It has nothing to do with fluidity. In fact, it is quite rigid. Same with D. It's an integral protein that is quite rigid. And A is a little molecule here called a carbohydrate chain, and it is on this protein here, so we call it a glycoprotein, and it has many functions, but none of them are related to making the membrane fluid. It is simply these, I'll show you here another picture, these fatty acid tails. They're very weak, like they're like little strings, and when you they make the membrane quite weak because they're very jiggly. Okay, basically that's another word you can use. So the answer is B. Okay, which is essential feature of Davson-Danieli model of membrane structure? So again, we saw this in part 2. Let me quickly show you. You need to know about three models of the membrane over the years. So the first model was the Garter-Grendel model, which was proposed in 1925, and this model showed that the membrane looks like the picture here. Right, a phospholipid bilayer. We know it's more complicated than that. So this was the first model, 1925. After that, 10 years later, came Davson-Danieli model, 1935. This one proposed that proteins, there's a layer of protein on the top and the bottom of this phospholipid layer, right? So that's what the Davson-Danieli model is. So let's see. So what is the feature of the Davson-Danieli model? A phospholipid monolayer. Nope, it should be a bilayer because it's still a bilayer, right? It's not just one layer. So it's definitely not going to be A. What about B? A layer of protein sandwiched between two layers of lipid. No, it's a layer of lipids sandwiched between two proteins. So they're using the wrong order here. So it's not B. C, a phospholipid bilayer, that's right, with intrinsic and extrinsic proteins. No, it's not this one. This right here is describing this model. It's describing the currently accepted model called the Singer-Nicolson model, right? Because this model, proposed in 1972, has intrinsic and peripheral proteins, unlike the Davson-Danieli model. So it's got to be D, a layer of lipid, right? The phospholipid bilayer sandwiched between two layers of protein. See, that sounds right. So it's going to be D. So you can see how important it is to actually know about these different models over the years, because they can pop up pretty easily on multiple choice questions. Again here, see another question. The Davson-Danieli model of a membrane structure proposed that membranes were composed of a phospholipid bilayer that lies between two layers of globular proteins. Like this, just like the image I showed you here. What evidence supported this model? So what made scientists believe this model was accurate? Because we know nowadays this model is not accurate, right? It's the Singer and Nicolson model that's the accurate model. This one is wrong. So what evidence supported this idea? Basically, I'll show you before we look at the options. There's a scientist who took an electron microscope, okay, and he looked at the membrane of the cell, and this is what he saw. And he saw two dark lines, a dark line here and a dark line here. And he thought to himself, and then a light line in the middle. So he thought to himself, this light line represents the phospholipid bilayer, and this dark line represents the protein layer on the outside and the inside. So that's what he thought. They used this electron microscope, and based on that, they came up with this structure. But it was later proved to be wrong. So is it A, an electron micrograph that showed two dark lines with a lighter band in between? See, exactly, it's this answer. It's going to be A. So it's not going to be B, C, or D. So B, freeze fracture electron microscopy. No, this was a technique. This was, in fact, the technique used, freeze etching, to disprove this idea, not to prove this idea. They used this idea to prove that Davson-Danieli model was wrong and the Singer-Nicolson model was right. Okay, so these are just wrong. So it's if you know, you just need to know about the basic reason why they thought this reason was right, and you can just think of this idea here. Which statement applies to cholesterol? So we learned about cholesterol before. This is this little molecule, this four hexagon shaped structure that is inside, mixed in between here in the phospholipids. And what applies to this molecule? It is hydrophobic and found on the outside of the phospholipid bilayer. Is that true? No, right? It's found on the inside, right here. It is hydrophilic and found inside the phospholipid bilayer. Okay, it is not hydrophilic. It is hydrophobic. Hydrophilic means loving water. We know it's in here, inside the phospholipid bilayer, because it dislikes water. It doesn't want to be in contact with the water. That's why it's hiding inside this part here with the fatty acid tails. So hydrophilic is the wrong part of this sentence. C, it impacts membrane fluidity. Yeah, that's true. We know that this molecule helps make this membrane not too rigid and not too fluid. It helps separate these phospholipids so that they don't bind too tightly together to make the membrane too rigid, and they also prevent them from floating around randomly. So if they're very weak and very fragile, it keeps them together. So it has a very important role in controlling membrane fluidity. So the answer is going to be C. It is transported in association with glucose in the blood. So we know this is not true, because think about it. Cholesterol is hydrophobic, meaning it doesn't like water. Your blood is mainly made up of water. So do you think it can be transported inside your blood? No, right? It struggles to be transported inside your blood because it dislikes water, so it doesn't want to be in your blood. So that's why D is incorrect. So it's going to be C. So that's it for these videos on membrane structure. You can do more, just go find more past papers, or just email me. I'm pretty sure you can find it somewhere on my YouTube channel. Just email me, and I can send you more questions and more questions. So the more you can do, I assure you, the better. The more confident you'll get.